All right, we're going to resume here with a talk by Russ Poldrack from Stanford University. I get to hear this talk once a year here at NeuroAcademy for every NeuroAcademy that we've done. I think this is probably the 9th time that you've given this. So it turns out reproducibility is still a problem, and we'll hear all about it from Russ. Thanks, Russ.
Thanks. Thanks for having me back once again. It's always nice to see you, and hopefully I have a chance to, I'm around today and tomorrow, so hopefully I have a chance to meet a lot of you while I'm here. Yeah, so I'm going to kind of introduce the big picture of like why it's important to do the things that you're going to be doing, and why it's important to learn the stuff you're going to be learning about over the next 2 weeks. And I'm going to kind of set up the problem. Where are we at? With kind of thinking about, the money that goes into the research that we do. So this is, I haven't refreshed this in a couple of years, but a few years ago, NIH was putting roughly, about $500 million into research that mentioned fMRI in its abstract on the NIH reporter system. And if you look at, what a word cloud of all the abstracts for those grants, you see what do people think they're getting out of fMRI research? Well, they think they're telling us something about brain systems in clinical patients and what the mechanisms might be of psychiatric disorders and how treatment might work and how things might change over time. And so the question is, like, are the taxpayers who are putting all this money in, are they getting their money's worth? And so you would hope that that we can tell them something like, what's going on in the brains of people who are diagnosed with depression. So this is a meta-analysis. Mueller et al. Published a kind of a re-meta-analysis looking at several previous meta-analyses that had looked at studies we call case control studies comparing people diagnosed with depression and people without a diagnosis. And each of these 99 studies had basically said, hey, look, we found a place in the brain where there is differential activity on some kind of fMRI task in people who are depressed and not depressed. And previous meta-analyses had said, hey, look, when we put these studies together, we can find that there are regions that are systematically, consistently different between people with depression and not. Mueller et al., who were kind of the world's best imaging meta-analysis people, fixed some of the problems of the previous meta-analyses, redid this, and basically found no consistent differences across. So 99 studies had told us there's a difference, but when you put them all together, you basically get nothing. That seems problematic. Here's maybe an even more glaring example of the kind of problems that can exist. So NIH had a press release about this paper that had come out saying that researchers use machine learning to analyze brain images and identify people with suicidal thoughts. If validated in larger groups of people, the approach could be used to assess risk and monitor response to treatments. And here's the picture from the press release that Carnegie Mellon had put out and that they reused, showing the brain activation pattern, for suicide-related words in people who had expressed suicidal, who had made a suicide attempt versus not. And they ended up getting almost $4 million from NIMH to follow up on this work. This work was later retracted 6 years later when some other researchers looked at what had been done and realized that they had made what is almost certainly the most common mistake that people make when they're applying machine learning techniques to, to almost any question, which is that they basically reused. You're supposed to keep the data that you train the model on and the data that you test the model on completely separate. And they did something that basically leaked data from training to test. We call it leakage. And when you get rid of that, basically the result goes away. They had to retract the paper. So this is a mess, right? It's not clear it's any worse of a mess than any other part of science, but it's our mess because it's the part of science that we play in. So how do we fix it? So I like this quote from Brandeis, the U.S. Supreme Court Justice. Sunlight is said to be the disinfectant, the best disinfectant. And that kind of underlies a lot of the work that we do to try to make science better, which is really to make it more transparent. So we've done a lot of work that I'm not going to go into any detail today in trying to develop this kind of like open ecosystem for for neuroimaging data analysis. You'll learn about a number of these tools. You'll learn about BIDS later in the week from Dora. You'll learn about some of the other tools from other people. And so hopefully you'll have a chance to try some of these tools out and see them in action. I'm not going to focus on them today. What I want to focus on is kind of the more fundamental structural types of questions. And I like to think about science as a design problem, right? Science is something people do, and whenever people interact with systems, there are, kind of things that people are likely to do that are good, and things are likely to do that are bad. Does anybody know what this thing is? It's a commercial paper cutter. You put a ream of paper in, and you punch a button, and it'll chop it, in half. So it has two green buttons right here. Why do you think that machine has two green buttons? What do you think is the thing that people might do if it didn't have two green buttons? They would hold the paper in and press the one button and cut off their fingers, right? So this is basically predicting the stupid things that people will do and trying to prevent them, right? And that's what design is all about, right? Like you go read any of the books on design, it's about like designing things so that people will do the things you want them to do and avoid the things that you don't want them to do. And it turns out that people are really good at fooling themselves. And so when we think about designing how we're going to build scientific systems, we need to think about how do we prevent people from fooling themselves. And so a lot of what I'm going to do today is talking about all the ways that we all, tend to try to fool ourselves. And so another idea that comes from behavioral economics is this idea of choice architecture, that whenever we go into a system where humans have to make decisions, some decisions are afforded, some, others are not, right? And so, for example, if you use software, if you've ever used any kind of data analysis software, there are very often default settings, right? There's a default threshold for something or a default number of something else. And if you don't know any better, you're very likely to use those defaults, right? And so the question is, are those defaults good or not? You can also try to predict the kinds of errors that people can make and give them feedback about what's going on to try to help prevent. Hello. If you're not muted, please mute yourself. Okay, so let's talk about some of the ways that things can go wrong. This is a great paper. If you haven't read it, it's a classic, now almost 20 years old, by John Ioannidis, called "Why Most Published Research Results Are False." And he tries to make the case that exactly what the title says. And one of the things he does is he talks about a number of the different features of a scientific study that could make it more or less likely to be true. And he says the smaller the study, this is one of the biggest ones, the smaller the studies performed in the field, the less likely they are to be true. And it turns out that most studies done in neuroscience generally, not just neuroimaging, but neuroscience more generally, are very badly powered. And in fact, in biomedical science more generally, not just neuroscience, most studies are badly powered, right? So statistical power means if there's a true effect, how likely are you to find it? If you're doing a study with 10% power, that means you have even if the effect is there, you'd have to run your study on average 10 times to find it once. The other 9 are going to give you a null result. You might say, well, that's not so bad. It's not going to make us fool ourselves. But it turns out that it can, because as Ioannidis showed in his paper, when your power is low, the likelihood that any positive finding that you get is true is also low. So think about this. I like thought experiments. So let's say that I'm a physicist and I'm doing some experiments, but unbeknownst to me, my detector is broken. So it's just giving me random noise. So I go do 100 experiments, and let's say if my P is less than 0.05 on my statistical test, I'm going to find 5 positive results, right? How many of those positive results are true? None of them. So my positive predictive value, the likelihood that a positive finding is true, is 0, right? And as I fix my detector, I'm still going to get those 5% of false positives, but I'm going to start getting more and more true positives, assuming there's an effect there. And so the likelihood that a positive result is true goes up as my power of my statistical tool is increasing. We also overestimate the effect sizes with smaller studies, right? Because with low power, the only effects you can find are really big ones. And so whenever you find an effect in a small study, it's almost certainly going to be a much bigger effect than the true effect size. We call this the winner's curse. Kate Button's paper from 2013 does a great overview of many of these issues. Another thing that we see is that with small samples, the effect that you estimate is highly variable. This is just a simulation from Felix Schoenbrod and Marco Perugini that showed that this is an example of like increasing your sample size. Each of the tiny lines here is basically just adding a data point and recomputing the correlation. This is just with simulated data where the true correlation is 0.26. But the idea is that like with small samples, when the true correlation is 0.26, you can see a correlation anywhere from say 0.6 something to minus 0.25 something. So with small samples, our statistical estimates are just all over the place. And if you're familiar with this paper by Marek et al. That came out a couple of years ago now, it basically just, I was a reviewer on this paper, and in my review I said, you know, it's an interesting paper, but it doesn't tell us anything we didn't know from those, you know, those simulations done almost 10 years ago. Nonetheless, I thought it should be published because I thought it was an important point to make. The point they make is simply that with small samples, your effect size estimates are one of the points they make. Your effect size estimates are going to be wildly variable. It's definitely a paper worth reading for a number of points that they make, but that was an important one. So it turns out that we know that this can cause people to fool themselves on a pretty grand scale. Back in the early 2000s, it became possible to measure genetic variants on a number of different genes that people were interested in, and then compare people, you know, with one version versus another version of that genetic variant. A popular variant that was this thing called the brain-derived neurotrophic factor, BDNF. It turns out that there's a genetic variant that causes like some people to have, you know, like more BDNF than others. And so what people started doing was getting a sample of people, taking their genotype, and then, for example, comparing how big is their hippocampus. We know that BDNF is expressed in the hippocampus. There's a good reason to think it might be related to hippocampal volume. And so early studies in the early 2000s, so this is showing you the effect size on the y-axis, the publication date on the x-axis, and the sample size is relevant to the size of the dot, right? So this is, I think, like 20 something, maybe like a very small sample. These are hundreds with the big circles. So you see what happens over time. Early on, you have very large effects coming from very small samples. All positive, right? You're not seeing anybody reporting negative findings here because you don't get a science paper for reporting a negative finding. And over time, the sample sizes get bigger, the effect sizes get smaller, and we now know that there's absolutely no effect. Because, you know, now that people have realized that these, what we call candidate gene association studies, were basically a mill for irreproducible results, they've moved to doing very large genome-wide association studies with tens of thousands of subjects. And this is from a paper that did that basically said, you know, previously identified polymorphisms associated with hippocampal volume showed little association in our analysis of, say, about 20,000 people. So basically, we know now that those just don't replicate. This phenomenon is seen really broadly. It's known as the decline effect. Over time, as studies get bigger, sample sizes get smaller, and it's probably due both to you know, problematic research practices, right? Like playing with your analysis until you get a big effect. And it's also due to publication bias, the fact that nobody wants to try to publish a negative finding because journals often publish them. So how good are we in terms of statistical power in fMRI? I apologize to those of you who aren't, you know, who don't do fMRI. It's the world I work in, and so it's my focus today. I think that almost all of the points that I've made, you know, this was in fact structural MRI. All the points that I make today are relevant to any kind of, you know, neuroscience analysis. I'm just focusing on the fMRI version. So we did an analysis a while ago, and this is an updated version with data up through 2023, basically showing how have sample sizes and statistical power changed over time. And so the median study, you can see the sample sizes have gone up. This is a log scale, so it's actually going up faster than it looks. The median study in 2023 was about 33 subjects per group. And it had power to find a single activation with 200 voxels. We had to pick some arbitrary effect that we're looking for. Let's say there's an activation we want to find with 200 voxels. We have power of about 0.7 an effect size of about 0.7 is the effect size that we have sufficient power to find. So we want to make sure whenever we're trying to design a study, we want to make sure that we have enough power to find an effect of interest, right? If I say I'm going to know, whether 4-year-olds are shorter than 20-year-olds, I don't need a very big sample to be able to say with confidence that 4-year-olds are shorter than 20-year-olds, right? If I want to ask whether 18-year-olds are shorter than 19-year-olds, I'm going to need a very big sample because that's a very small effect compared to the difference between 4-year-olds and 20-year-olds. So I need a much bigger sample to say with any confidence there's a difference between 18 and 19-year-olds. So the question is, do we actually have sufficient power in our fMRI studies to find, kind of interesting effects? And so the problem is you can't take published effect sizes because they all suffer from that winner's curse. You have to go find realistic effect sizes. So we do that by taking an independent data set, the Human Connectome Project data set that you already heard about, and doing analyses of a bunch of the different tasks that they have in that data set. They had already written down what the regions of interest were for each of the tasks as they were being designed. And then we also used a tool called Neurosynth that I think you'll hear about, where you can basically go in and ask, for example, for the motor task, what are the regions that are activated across, any task that mentions motor, and basically identify the voxels that we're really sure should be active if it's, say, a motor task or a working memory task or whatever the task might be. And then we put all those together to estimate the effect sizes. So first, look at the thing on the left here. This is saying basically, it's what we call percent signal changes. How many percent does the signal go up when the person does the task versus just resting? And in general, the biggest percent signal change you'll ever expect is on the order of like maybe 4 to 5%. If I show you flashing stimuli and I look in your visual cortex, if I move your hand really fast and I look in your motor cortex, we'll get roughly 4% signal change. Most interesting tasks, like the stuff that my lab studies, usually an order of magnitude smaller, like certainly much less than 0.5% usually. And so the tasks, because these are pretty whopping tasks that were used in the Human Connectome Project, they all show roughly at least 2% signal change. The motor one gets up above 4%. So these are really big effects, right? Most interesting cognitive studies would not be looking at effects this big. But the question is, how much statistical power do we have? And statistical power looks at, because we're interested in looking at, the group average, we have to look at how big is the effect with respect to the variability across people. So you're taking this number, but then dividing, some measure of variability across people. And we often quantify, there's these kind of arbitrary cutoffs for small, medium, and large effects in psychology. And so you can see that the biggest effect, that this whopping effect, is kind of like a medium to large effect size. A lot of them that are in the 2% range are, even down here in the small to medium effect size range. So what this is saying is the average study published in 2023 was only sufficiently powered to find activation for things like moving your hand, right? It's certainly not powered to find any kind of interesting kind of cognitive neuroscience effect. So we have a power problem. In fact, if you look at the studies from that meta-analysis, you can see that that's probably a big part of the issue there, that many of these studies had what I call an insufficient grossly insufficient sample size. Based on this, this is guidance from, there's a paper published in 2011 called False Positive Psychology, really great paper about, it's actually the paper I think that coined the term p-hacking. And they give a bunch of guidelines. And one of their guidelines is that it never makes sense to collect less than 20 subjects per group, unless there's some really strong justification in terms of the difficulty of getting those samples, because you would never do a power analysis that says 20 is enough. at least not for almost any interesting scientific effect. And so much more often, they mention this, they're more likely to reflect interim data analysis and a flexible termination rule. What does that mean? That means we run 12 subjects and we do our statistics and we see if there's an effect. And if there is, we stop. And if there isn't, we keep going. And we do that after every few more subjects. And whenever we find an effect, then we stop. And we know that that greatly inflates the error rates in studies. Now, it turns out that the same issues are present in machine learning studies. Machine learning does not save you. In fact, it might even make things worse. Gael Varacqua has a great paper from a few years ago called cross-validation failure, which showed basically how small samples can lead to high variability in predictive accuracy when you apply machine learning methods. And he also showed this decline effect also happens for machine learning decoding results. So this is basically showing how the reported predictive power, like how this is for a bunch of different diagnoses. How well can you predict for a person who was held out, which group they were in? The smaller the sample size, the better you do. Clearly, that's because you're probably taking advantage of the fact that there's a lot more variability. So like some studies would publish, 100% accuracy at doing some sort of psychiatric diagnosis, right? That's clearly not a reasonable number. Anytime when you're doing machine learning, anytime you get 100% accuracy, unless it's on a problem that's just, mind-numbingly boring, you've almost certainly done something wrong, and they almost certainly did something wrong. Questions? I should say, feel free to raise your hand and stop me anytime. I think we have till noon, right? Cool. So yeah, please feel free to stop me. So one, when you become a professor, you've got people invite you to go give talks, and many of you might have had this experience where you go to lunch with a speaker, before the talk or after the talk. And one of the questions that when I would go give talks about this stuff, back in, the last 10 years, trainees would often ask, what, you just told me that we need these huge samples and, blah, 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 we need to do all this stuff. How am I supposed to go do a PhD, right? I can't go get a sample of 20,000 people to ask the question that I want to ask. What am I supposed to do? So I wrote a paper that you can have a look at called The Cost of Reproducibility, back in 2019, that basically tried to address this question that had come up so often in these talks, one way to phrase what I say in the paper is basically just a kind of a rephrasing of what Nancy Kamwitcher said, which is if you can't answer the question you love the question you can answer, right? So basically, you have to pivot, right? If the thing you want to do requires 20,000 subjects and you can only get 100, then you shouldn't go do 100. You shouldn't go just get 100 and do the thing that's underpowered, right? What you should do is figure out a way to pivot to address the question or a similar question or a related question or another question in a way that's going to be rigorous. And so how do you do that? You might collaborate, right? So what some people in some fields in genomics, people now work across large consortia, right? They all share data together. And, in other cases, multiple groups might come together. If they're all collecting, very precious samples that are hard to get a lot of, rather than publishing a bunch of papers with very small samples, they can come together and publish a paper with larger sample. Using shared data, as you heard, and I think I'll mention, right, there's this sort of a golden age of data sharing, right? There's so much data available online. The problem really becomes like, how do you figure out what's the right data set to work with and how do you process it so that you can use it? Another thing you can do is focus more on theory, right? Or on computational modeling, on trying to figure out how can you ask, questions in a different way. So I'm happy to just kind of, come back to any of those points in our discussion later if there are any questions. Okay, so back to Ioannidis's points. The greater the flexibility in designs, definitions, outcomes, and analytical modes in a scientific field, the less likely the findings are to be true. So that basically means that To the degree that everybody's analyzing data in a different way, and there's no constraints on how you analyze data, that's a problem. So we, in this paper we published a few years ago, we went through just for one of the software packages in fMRI and looked at, what are all the choices that one has to make in running an fMRI analysis, processing, and then statistically analyzing data, and how many plausible options are there at each step. And so that means that there's, one could reasonably choose each of those things. Put those all together, you get upwards of 69,000 different plausible workflows that one could use. That's only for FSL, right? Each software package also has all those. And if you start mixing and matching software packages, then it gets even bigger. And it turns out that people actually do all sorts of things. Josh Karp did an analysis a few years ago where he just looked across papers at, how different are the workflows in the published fMRI literature. And he found that there's nearly as many unique analysis pipelines as there were studies in the sample, which is 241 studies. So almost everybody did something differently. Even within the lab, groups rarely use exactly the same workflow across papers. He also showed in another paper that this actually matters for the results. So here he took one of the data sets that we had shared and reanalyzed it across around 6,000 different workflows, just varying, different things, and then asked, how do the statistical results differ? So what you're seeing here on the left is just the average activation map across all of those different analyses. Unfortunately, it looked like the thing we had published, so that was good. And it looks like a reasonable activation map, but the thing on the right is what's really disturbing, which is the range of the Z statistic. So this is what's the difference in standard deviations between the highest and lowest Z statistic. And you can see in some regions, there's a difference on the order of like 8 standard deviations, huge differences in the statistical results between different pipelines. Machine learning can make this worse. There's this great paper called I Tried a Bunch of Things, which basically shows how you can get, seemingly good results from complete noise by basically just using machine learning to find better, better algorithms on your data, basically overfitting to the data. It's also not just fMRI. There's this great paper by Luck and Gastelum, which went into how things that people do in the ERP literature are almost certain to raise the false positive rates above 50%. So not just fMRI. Now, one of the things you can do that we know can help improve the rigor of data analysis is pre-registration, which basically just means writing down everything that you plan to do, preferably including, the code that you plan to use for your analyses, and putting that in a place where it basically can't be changed and it's time-stamped, right? So you and everybody else know exactly when you pre-registered it. Preferably, you want to do this before you collect the data, but even if you're using pre-existing data, you can still do this. And even because I think, people often think of pre-registration. How many of you have heard of pre-registration? Okay, good. People often think of pre-registration as like something that, the world can use to tell whether a person is like, really telling the truth or something like that. I don't think that's the way to think about pre-registration. I think about pre-registration as a way to keep me honest with myself, right? Because it's very easy to start looking at your data and think, oh yeah, I really meant to do X, when if you had asked me before I saw the data, I wouldn't tell you I meant to do X. Like X seems like in hindsight, things can seem very obvious when in foresight, they would not have seemed obvious at all. So pre-registration really helps you know what you were thinking before you ever touched the data. And it's important to say, like, every paper that we've ever published where we had pre-registered analyses, we had to deviate in some way from what we pre-registered. fMRI data are complex. You almost always run into something you didn't expect. You write in the paper, we deviated from the pre-registration in these ways. And reviewers can occasionally be prickly, but in general, it's not been a problem. So this is a pretty amazing result, I think. So this is from the clinical trials world. The National Heart, Lung, and Blood Institute in the US in 2000 started requiring people running clinical trials to pre-register their design, and importantly, to pre-register what they call their primary outcome. What's the thing that you're trying to affect with your treatment? Is it the number of people who die, the number of people who have a heart attack, some other thing that you're trying to treat. And so they just basically said, what's the thing you're trying to measure? Because I think there was an intuition that what people often do is they measure a lot of things, and then they look at all those things and find the one that had a significant effect to the treatment, and then write the paper as if that's the thing they predicted an effect on. So prior to 2000, so basically this is relative risk. So below 1 means the treatment is effective. And that's why you see a bunch of dots before 2000 with little plus signs, which means they found a significant positive result. Only one that had a really negative result, and it wasn't even significant. Look at what happened in 2000. Suddenly, almost all of the results are null. It's not as if drugs stopped working in the year 2000, right? What happened was that people who were running these trials could no longer move the goalpost so that they could, claim to have a significant result when they really didn't, at least on their primary outcome. Now, pre-registration can prevent p-hacking, right? By basically fixing what you plan to do. Obviously, it doesn't, 20 different groups can still pre-register 20 different analysis pipelines, right? So it doesn't address the variability problem. So we wanted to know, really in the wild, if you give people data to analyze, how differently are they going to analyze it, and how big of a impact does this have? And so we got involved in this study we call the NARPS study, Neuroimaging Analysis, Replication, and Prediction Study, led by Tom Schoenberg and Rotem Nazair in Israel. And so what we did was we collected an fMRI data set on 108 subjects, using two different versions of a task that we had used before in our lab. The task doesn't really matter, but it was a decision-making task. And then we basically recruited people to analyze the data. We went out on social media, went to conferences, and basically said, hey, we have this data set. We want groups of up to 3 people to analyze it. We'll give you a few months. We ended up giving them a little over 3 months. And we want you to give us back a set of answers on particular questions. 9 hypothesis tests we want them to just give us yes, no answers on, which were of the form, is there significant activation in some region of the brain for some aspect of the task? So for example, in the study, there were some gambles with low gains, some with high gains. And we want to know, is there a difference in activity in the ventromedial prefrontal cortex that relates to how big the gain is, or how big the loss is in the gamble? And we told them basically, analyze this as if it were your own data in your lab. The only thing we required is it had to be a whole brain analysis. We got back results from 70 teams, and they gave us a bunch more than just those 9 hypothesis test outcomes, but that's what I'm going to focus on. We had them. So there's now a checklist that, if you're writing an fMRI paper, you should be familiar with, called the Cobitus checklist, which basically lays out a set of reporting practices for fMRI papers. We had them give us a detailed description of what they had done. And we basically found that no 2 teams used an identical workflow of the 70. But it also was, even with that long, 10 to 20 page description of what they did, it was often hard to know exactly what they had done. We had to go back and ask for code or other files to really see what they had done. So here are the results. The y-axis is the proportion of teams saying yes on each of the 9 hypotheses. They're ordered, they're ranked by that outcome. So this means that about 85% of the teams said yes, there is an effect in that region for that aspect of the task. For these 3, nearly all the teams said no, there's no activation for that aspect of the task. But then for these 5 of the 9 hypotheses, the 20 to 40% of the group said yes. Same data, and no incentive to p-hack here, right? There was no incentive for them to have yes or no answers. We just wanted to know, what do you think the truth is? And even with no incentive to p-hack, we get this amount of variability across teams. 33 different patterns of yes-no outcomes across the 70 teams. And for any particular hypothesis, there's at least 4 workflows that can give you a positive answer. If you look at the voxel-wise results, it's kind of equally variable. This is showing at each voxel, what's the proportion of teams. This is for the best of those hypotheses. What's the proportion of voxels across, the proportion of teams showing a significant effect at each voxel. So basically this says that no voxel has more than 76% of teams saying yes. Skip that. So when we also looked at the underlying statistical maps, not thresholded, but just the whole brain statistical maps. And what you see here is a correlation matrix color-coded by where each row by column is one of those 70 teams. And what should pop out at you is that there's this big group of teams up here that are highly correlated with one another in terms of the underlying statistical maps, the statistical values at each voxel. But some of those, even though they're highly correlated, this is red, green here means yes or no on that hypothesis. So teams could have very similar statistical maps, but still give you different yes, no answers. Maybe more striking is the fact that there's this one set of teams here, I think it was 8 or 9 teams, that are negatively correlated with all these other teams. The blue here means that these are all correlated with themselves, but negatively correlated with the others. And if you look at the average maps for those teams, they look like the opposite of that map, right? Whereas these had red here in the ventromedial prefrontal cortex, these have blue. If you've looked at much task fMRI, you're going to recognize that this just looks like a task versus baseline activation, right? This is the default mode deactivation that we always see. And we dug in to figure out what they had done. And it turns out that at least for 7 of the teams where we could get good information, they basically not specified their statistical model correctly. They basically hadn't put an intercept into the regression, which is you would never do if you're doing regression, on simple data. It's really easy to do in fMRI with these complex designs. Nonetheless, if we put the data all together, even with those bad teams in there, if we put the data all together, we can get consistent results across all the teams, right? So this shows for several of the different hypotheses. This is number 5 here is the one that the teams all agreed upon, like the 85%. You can see that you get, really nice results across, when you combine all the teams. And it would be better if we threw out the 7 who didn't do the model correctly. So we think that these types of results suggest various ways to try to address reproducibility problems. One is doing what we did here, which we call a multi-analyst study. And there's a number of groups that are now trying to do this. We wrote a consensus paper on basically how to conduct and report multi-analyst studies. There's other things that people have done in other fields of science. So in physics, people do this thing called blind analysis, where the person who gets the data doesn't necessarily know what exactly they're looking at, or some parts of the data have been removed. There's a great Netflix thing, or it's on Netflix, about the people who made the first images of a black hole. I forget what there's this kind of observatory with a bunch of different telescopes. And they basically had 2 groups who could not talk to each other, who were both analyzing at the same time. And they come together at the end and they show their images at the same time. And they look pretty much the same, which was cool. But that's like in physics, it's very standard to do this sort of blind analysis, because they found that when they didn't do that, they would see bias in the results. Another thing one can do that we've started doing in my lab is basically, before you ever touch the real data, simulate data and develop your pipelines on the simulated data, so that you can be sure that if you simulate it to have the signal you're looking for, you can find that signal. If you simulate it to have no signal, then you find no signal. This is hard to do with fMRI. It's easier to do with behavioral data and kind of smaller studies. So I think we have a ways to go in terms of fMRI simulation. There are tools, but they're just kind of janky. Okay, so that's kind of a break point in the talk, because we're going to move now towards talking about writing code. So maybe why don't I give you an opportunity to ask any questions you might have.
Simulating, would you say it makes sense to collect pilot data of say 2 and 3 participants to get data that is structurally exciting that and double pipeline? I mean, my theory it would be that today your analysis to those 2 or 3 subjects would be
Yeah. So the question for those of you on Zoom was basically, would it make sense, given the difficulties in simulating fMRI data, would it make sense to basically collect a small pilot sample of, say, a few subjects, and then use those subjects' data to develop the pipelines, and then, you know, kind of apply those pipelines to the full sample. I think that's a very legitimate strategy. It's going to work, you know, for some types of studies, it's going to work better than others. If you're doing stuff that really requires, you know, combining across subjects, then doing that with a small sample of 3 is going to be challenging. But, you know, what you could do is take those 3 and maybe, like, you know, add some noise to them, or use them as the basis for like a simpler simulation. So I think that's probably one of the best strategies to try to address that. Yeah.
Got some questions from people on Zoom with their hand raised. However, I think just today. So for those of you on Zoom, just today, this morning, we won't be able to hear you in the room, I don't think. Oh, okay, so I think Angelina, you had your hand up. Do you want to try to ask your question?
Sure. Can you hear me well?
Yeah.
Yes, thank you very much for a very, very fascinating talk. I'm a third-year PhD student, so of course an ECR, and everything that you mentioned is absolutely valid. And I think all of us would like to live in a world where these principles can be actually enforced. But I can't help it but think this is just not feasible right now. Why do you think pre-registration is not mandatory in science yet?
Or
something similar?
Yeah. No, I think that's a great question. And you know, there's a lot of answers. Like, you know, science is ultimately a social enterprise, right? And it relies on people, you know, making decisions and people having the right incentives. And a big part of why it isn't required right now is that the incentive structures are not aligned with the goals of pre-registration, right? That people aren't incentivized to do good science. They're incentivized to publish papers in glossy magazines like Science and Nature, because that's what gets you a job, right? And so until we can align the incentives, people, you know, it's going to be very difficult to convince, you know, the masses in science to do things well. And particularly, you know, there's an overlay here, which is the big decisions are unfortunately not made by people in their 20s and 30s. They're made by people in their 50s and 60s and 70s, most of whom got where they are by doing all the things that I'm now saying one shouldn't do, right? And so it you know, obviously I and a number of people from my cohort have said, hey, look, we need to change what we're doing. But a lot of people find that, a lot of, you know, people of my vintage and older find that threatening, right? Because you're basically saying, you're kind of making them, you know, admit that a lot of what they might have done in the past is less robust than they thought it was. And so I think that it's a real challenge. Now, there's a lot of work going on to try to make these kind of changes. We're involved in a national group in the US called Helios, Higher Education Leadership Initiative on Open Scholarship, which is basically it's like, you know, over 100 colleges and universities trying to make structural change. But ultimately, it's really hard to do that, you know, because it's the hiring and tenure and promotion decisions are being made mostly by older faculty. And so many of you might have heard the phrase science progresses one funeral at a time. I think this is in part, you know, what has to happen. Somebody once asked me, like, if you know, if you were given all the money in the world and could do one thing to like improve science, right, what would you do? And I said, I would force everybody over 50 to retire, which obviously isn't going to happen. I mean, and that would force me to retire too. So I hope that addresses your question. I think it's ultimately a really hard question. The reason that journals don't require it for the most part is because journals are mostly in the business of making money. And if they require something that a lot of people don't want to do, then people aren't going to submit to their journal, and their number of papers they publish are going to go down, and their impact factor, which they care about, is going to go down. And so like journals, many journals, like I'm one of the senior associate editors at Psychological Science. And even though we don't require pre-registration, but when we look at papers to determine whether to send them out for review, we definitely look closely. And if it is not pre-registered, then we give it a much closer, screening before we decide to send it out. And we also give badges for papers that were pre-registered. So there's movement. It's slow. But, you know, as the, what does it say, as the inmates take over the asylum, then things will get better, I hope.
Thank you.
Yep. Were there other Zoom questions?
Yeah. Alex.
I think you said Alex. Hey, along the same vein, really great talk. I was just going to ask, where do you think the needle is on the 70 teams of researchers, you know, if they had chosen to write one small sample size paper instead of participating in the study, you know, where does that help their career? Are we getting towards, you know, were we able to give those researchers in that team study their due for their career progression?
That's a great question. I don't know is the honest answer. I mean, I think it's slow. You know, other fields have figured out how to do this. Like in physics, you regularly see papers with hundreds of authors, right? And people get career credit for being part of those papers with hundreds of authors. Similarly, in genomics, the paper that I showed, you know, from Jason Stein, it had maybe 100 papers in genomics, often have over 100 authors now, and people are getting jobs and getting credit for their work, not just the first author, but, you know, others as well. So I think other fields are figuring out how to do this, and we just need to take it more seriously. But in part, we need more examples of these papers to even see, you know, without having them, you know, without having us as faculty having to have the discussions about them. I don't even really know, because we haven't, when I think about like people whose CVs we've looked at for hiring, I don't remember there ever being a question about like how much credit they should get for having been part of a 100 author paper, because we just haven't seen any of those. Mohammed? We have one in the back of the room. Why don't we go to that and then we'll try to come back. So the question is, what do I think the role is for pre-registration if the research isn't hypothesis driven? I think that's a great question. So we certainly, you know, we do a lot of work that is really more discovery work rather than hypothesis driven work. We still try to pre-register whatever we can. And part of what we do in that work is to try to figure out like how are we, because, you know, the exploration part is not going to be confirmatory. You're not going to go do, you know, t-test and report, you know, p-values that or people are going to believe. But what you can at least do is write down what your strategy is going to be. How are you going to go forward and test that? Are you going to, for example, you know, one of the things that we always like to do when we do an exploratory study is we hold out part of the sample as our validation set, right? It's easier with behavioral work than with imaging because imaging is expensive. But if we're doing a behavioral study, we might collect 500 subjects, hold out, you know, 200 of them, fit our models and do all of our, you know, kind of our discovery work on the 300 and then validate it on the 200. So the question is, you know, anything you have that you can write down that will kind of just like, you know, help you figure out, you know, what you planned to do before you started doing it. Those are the kind of things that we think about pre-registering, even if it's not going to be like, what are the primary outcomes or something like that. It's for us, it's more like the strategy. Yeah.
So if somebody has data correct for the time scale, the depth of the universe, what does it matter if it's pre-registered or not? Which it is in a lot of science, just fMRI is a little bit earlier of a signal.
Yeah, no, I agree. If it's a really large effect, if it's like the person can talk versus can't, you know, isn't talking, right? Those types of effects, you know, I think are different, right? In the sense that like a much smaller sample, it's kind of like the 4-year-old versus 20-year-old height example, right? You don't need the same kind of sample size. I think it still could be useful to write down what the strategy is going to be. But I agree that, you know, if it's compared to like, you know, trying to find the Higgs boson at the Large Hadron Collider, that's a tiny signal, right? That requires a lot more work to do it rigorously. But I think that the strategy can still be used there. Is Mohammed back on? Sorry, do we have one more? Okay.
Hey there, Tomas, can you hear us?
Sorry, am I up?
Yep.
I was just gonna ask, in terms of, yeah, trying to make the most of small sample task fMRI studies, like, do you think doing Bayesian analyses could help somewhat? And why are there so few of them in the field as of now?
Sorry, I missed what kind of analyses?
Bayesian, yeah, just Bayesian
Oh.
analysis. Yeah.
So they're not going to save us. I think you can p-hack your way to success with Bayesian models just like you can with standard frequentist or likelihoodist approaches. I think that the thing that they're useful for is pointing people to think more about the evidence in favor of or against a hypothesis, as opposed to thinking about the p-value. Because in general, something like a Bayes factor is usually going to be a little bit more stringent than a p-value. That was the point of these moves to start requiring people to use p less than 0.005. So I think around the edges it can make a little bit of a difference. I don't think they're going to save us. I think, personally, I think that the bigger step that I think will have could be much more important is moving to out of sample validation. So taking the approaches used in machine learning and really saying, we're always going to have a validation set that we hold out, do all of our analyses on, and then kind of validate the result against that. Do we have time for another one in the room? Sorry?
If you have time.
Yeah. I'm good. Yeah.
As a PhD student, doing neuroimaging stuff, whenever I start taking it, really discouraged.
That's not my goal.
That also sounds scary. I'm wondering if you could expand on that a little bit, and if you have any examples of your own career where you pivoted a little bit.
Yeah. So certainly, I think the big pivot for me was about 10 years ago, like right around the time I moved to Stanford, right as the psychology replication crisis was kicking in. My lab had for 15 years been doing relatively small sample cognitive neuroscience studies. We had published a paper in Science with a correlation between brain and behavior of 0.8 something with 16 subjects. I look back at it and I'm like, wow, that's embarrassingly bad. And so the big pivot for me was we're not going to do these kind of small in studies anymore. And for a number of years, my lab didn't really collect much fMRI data at all. We've now moved, kind of the second half of that pivot is really moving towards collecting much more data per individual. And so now when we do imaging studies, we generally collect on the order of 10 hours of data per person. So much more like the way that people do it in the vision neuroscience world. We're also collecting relatively large samples. So the last study we did, it was 48 people, I think, each with 8 to 10 hours per person, collected over months because, we can't get people to come in and sit there for 10 hours. So those are the kinds of pivots that we have made to try to, do science differently. I think the other pivot was really moving much more towards technology development and infrastructure development. So we started developing tools for data sharing, like the Open Neuro platform. We started working on the brain imaging data structure to help people share data more effectively. We started developing software tools like fMRI prep. So that was probably the other, more than half of my group now is basically software developers or people doing data curation activities, things like that, not people doing basic science. Obviously, different people are in different positions to make different types of pivots. I think a common pivot for people interested in fMRI, but unable to ask the question they really want to ask is kind of double down on the behavioral side, on the cognitive side, or on to the degree that you're working in an area where kind of, computational models of behavior are prevalent, kind of digging into how can I better understand the behavior? Obviously, this is relevant for people in my lab. It might not be relevant for other labs, but how can you understand, are there are there other things you can become expert in, like on the behavioral side, that you can then translate if you, say go to a postdoc in another group where you might have access to larger data sets or working with a consortium, things like that. I don't know if that's helpful.
Was there a sense of?
No, I mean, I was a full professor already. So it's just, it's hard, it's very different for me to, like my experience is not really going to translate into an early career researcher because, I kind of do whatever I wanted to at that point, and I wasn't too worried about my career. So it's very different. Yeah. And I completely acknowledge that. So I'm slightly loath to give advice about how to go do things just because, it's hard to know what's going to work. And but yeah. Okay, why don't we go to the next part, and then if there's time at the end, we can obviously take more questions. Okay, so I'm going to pivot to talking about coding. Show of hands, how many of you have written code in the context of doing research? Okay, so the majority of people in the room are raising their hands. How many of you have been trained in software engineering? Okay, not a majority of hands. How many of you have ever written a test for your code? Okay, a plurality, but not a majority. So I have come to think that software errors are a significant threat to reproducibility. And this is just one example. Jeffrey Chang's a structural biologist who in the early 2000s had a set of papers in glossy magazines like Science about this thing called the ATP binding cassette transporter, and figure out like the protein structure, right? And he ended up having to retract 5 papers after they realized that there was a piece of software that they were using that had one sign change in it that ultimately caused the structure of the protein to be wrong, that they had estimated using their software. And some other researcher figured this out. They went in, looked at their code, realized that it was doing this thing, and had to go retract these papers, right? None of us wants to have to be in that position. Here's an example from our lab where we almost ended up in that position. It wouldn't have been quite as bad. It would have been one paper and it wouldn't have been, completely wrong. But this was a paper where, and I wrote a blog post about it a while ago, a paper where we had this shared data set with people doing one of 23 different cognitive tasks. And we wanted to ask, how well can we classify which task the person was doing out of those 23 just based on their brain activity pattern? We'd done this on a smaller scale before. We wanted to kind of ramp it up to this larger set of tasks. So I wrote this little bit of Python code to do that. And I did pretty well. I got 93% accuracy. If it had been 100% accuracy, I would have known I had done something wrong. But when I get 90% accuracy, I'm like, wow, that's pretty good. But I had someone who was a postdoc in my lab. He's now a computer science faculty at Stanford, Sammy Koyajo, look over the data, and he said, oh, no, you did something wrong. You forgot to put in this index thing. And so basically, it was misindexing the data. It was indexing it by subjects rather than splitting it by subjects rather than voxels. And the result was still well above chance. Chance here is like, less than 5%, but not 90 something. It's like 50 something. So even a small error involving like, 4 characters of text can have a pretty substantial impact on results. Here's another example from our lab where we made a mistake, and because we shared our code and the data were openly available, somebody else was actually able to have a look at it and find the error, and we were able to fix it before the result got published. So we had published a paper, we had put out a preprint about some issues that we had discovered with the behavioral data in the stop signal task in the ABCD study. And this is what our text in the paper said. 1326 of the 7,231 subjects had this gross violation of a particular assumption. And Sage Han, who was one of the software developers for the ABCD team, they were not happy about this paper because it was basically saying they had done a bunch of things wrong in developing the study. So they dug into our code, and this is if you're a Python coder, you probably don't even you don't have to be a Python coder to realize that that's probably a really non-robust piece of code, right? And in fact, the error had to do with one parenthesis being in the wrong place, and it caused the Boolean operation to give the wrong answer. And so we fortunately were able to fix it. It turned out that the number was 447 versus 1,326. It didn't change the overall story of the paper. The paper was still published. But this was an opportunity where, because we shared our data openly and were open to people, having a look and telling us we'd done something wrong and to fixing it, we had an opportunity to do that. So one of the undercurrents here, and something you're going to hear throughout the workshop, has to do with the importance of sharing resources and making them openly available. And I think that we as there's often this idea that if you make a mistake, that that's some kind of poor reflection on your character, on your quality as a scientist or something like that. And I think we need to move to a world where we realize that everybody makes mistakes. And our goal should be to find them and fix them, not to lay blame on people. So we actually, maybe I'll just mention something that we do in my lab that I've heard of other people doing similar things. There's a thing that people do in medical centers called the Morbidity and Mortality Conference, where they basically come together and it's in a blame-free environment, right? So it's not about so you want to figure out why did this bad thing happen? Why did this person die? Why did this person almost die? Why did we have a near miss? And figure out what happened in particular, not just what did this person do wrong, but how could the system have prevented this problem? So what we now, when we had another recent thing where we had done some analyses of the ABCD data set, and I forget where we were, and I think we had actually, the paper was submitted and had already gone through one round of review, and we were revising it, and the postdoc realized that he had made an error in something he was, I don't remember what the error was, something he was doing with the data that didn't really cause the conclusions to change at all, but it caused the numbers to be different. And so we sat down in our lab and walked through the code where the error was and tried to think through what could we have done to protect ourselves from this? And I think that's the way that we should think about absorbing errors, right? Not trying to hide from them. Obviously, again, this is one of these things that's really easy for me to say as somebody who has a full professor job and has a career. And I realize that this is a much harder thing to absorb for trainees. But I think it's, as you go forward, if you're building a lab, I think it's really important to have the people who work with you feel like if they make an error, the first thing they should do is come tell you, right? They shouldn't try to hide it from you until they can't, until it has to come out. That, you want to have this blame-free environment in your group. I think there's another overlay here, this thing that we call bug hacking, which is that, let's say that you make an error in the code. If that error causes your result to look really good, you're going to be really unlikely to dig in to try to see if there's an error there. Whereas if it causes your results to not come out the way that you expect, to be null or to be different, you're much more likely to go look for bugs, right? And that's the point of this particular PhD comics strip. And so I think we need to think about how do we protect ourselves from these types of these types of tendencies. And I wrote a blog post a while ago about something that we call the principle of assumed error, which is when in general, when we get a bad result, we often assume that we've done something wrong in our analysis. We should bring the same assumption to bear if we get a really good result, to try to dig in and figure out what did we do wrong. Just kind of double back. But when you're thinking about, you got something really cool, you want to write it up for a fancy journal, what are all the ways that could have been done wrong? And you want to double check and triple check and make sure that that you've looked hard. And sometimes it's going to find something that you don't like, but it's better to find it then than to find it after the paper has been published. So a lot of the stuff that you're going to learn here, is about, how to write code, and I think that, as you're doing that, you should think about how to write it in a way that's reproducible. What does that mean? means it should be readable. I'll come back and talk about that in a moment. It should be robust in the sense that it should, like, fail loudly, right? It shouldn't it shouldn't let you do a garbage in, garbage out kind of scenario, right? If you put garbage in, it should tell you, hey, I think that's garbage. It should be modular in the sense that it's small pieces, and that goes back to readability. And it should be well tested to the degree that you can write tests to make sure it's doing the right answer, doing the right thing. You should do that. And I like to think of coding as, I think the term software carpentry, if you're not familiar with software carpentry, they run a lot of, workshops. They have a lot of online materials about like how to improve your computational practices. And I like the analogy because coding really is like a craft, right? It's just like building stuff out of wood. It's building stuff out of, pieces of text that can do something in the world. And the only way you get better at it is to like keep doing it and to try to improve yourself, right? So read the literature about how to write better code. Use tools that can help you write better code. Now you can have, ChatGPT help you write better code. And I'll talk more about that tomorrow. But the important thing is like consistent and deliberate practice. It's going to, for almost all of you, it's going to be a central part of what you do in science. And so it's really important to learn to do it well, because otherwise you end up, like I've spent decades not doing it well. And now I have lots of really bad habits that I'm continually trying to break. So like forgetting to check things into Git and then realizing that I've done something and lost 20 minutes of work because I didn't have something checked into Git. So I think something that I've become much more kind of passionate about in the last few years is the importance of writing clean code. And part of the motivation for this is this idea that code is read much more often than it's written. That's a quote from the developer of Python. And when you think about who's reading the code, it's most often going to be you in the future, right? So if you write a piece of code, you come back to it in 8 months, you're probably not going to remember exactly what you were trying to do. So it's really important that you could kind of read the code. There are, like different languages have different principles. You're going to be focused on Python here. And so there's a set of, principles and standards around Python coding. There's also language independent principles. So I would suggest, I think of these are all great books. The first 3 are really long. I would start with The Art of Readable Code. There's a free PDF of it online if you go search for it. It's language independent, but it's basically about how to write code that's easily readable. And a lot of it, there's actually a whole book called Naming Things, which is all about how do you name things when you're writing code. It turns out, I think the subtitle of the book is like the hardest thing in computer science or in computer programming is naming things. And then this book by John Osterhout, A Philosophy of Software Design, talks a lot about like, how do you think about what we're trying to do when we build software? And has a lot of principles for thinking about, building more robust software. Now, people often think that the way to make code better is to add more comments, right? I often see people on Twitter saying, oh, I haven't shared my code yet because I want to go in and put a bunch of comments in. And when you read the literature on software development, it's clear that most people who have thought about this do not think that comments are a particularly useful thing in most cases. That comments are really important if there's something that you need the person reading the code to understand that wouldn't be obvious from the code itself. Like, why is it that you're doing a particular thing? Like, where's the, like you're using some particular default, where does that default come from? Is there a previous publication on it or something? So these are a couple of quotes. Like, clear and expressive code with few comments is far superior to cluttered and complex code with lots of comments, right? So what you want to do is write the code cleanly to begin with, or don't comment bad code, rewrite it, right? You want the code to be written as clearly as possible. Now, there's a lot of tools to help writing code, and hopefully you'll kind of have a chance to play with some of these. Some of them are what we call static analysis tools that basically just look at a piece of code and say, are there any errors? Do you have any syntax errors? Are there like any variables that haven't been defined that you're using or things like that because you like, had a typo in something? And you can set this up to run automatically. Like I use VS Code as my main code editor. I would highly recommend VS Code. But most development environments have some way to set it up such that every time you save the file, it'll run, like I use Flake 8, it'll run the tool and then give you a little report and it'll tell you, are there any errors? Are there any stylistic issues? And then you can try to fix those. There's also tools you can use to basically improve your code to make not necessarily it's not going to change the names of variables and make them better named, but it will at least change the formatting of the code if you're, for example, you don't have spaces in the right places and stuff like that. So tools like Black or Blue for Python that can take a piece of code and then basically just make it prettier, make it more compliant with the coding standards. Code review, I think, is a really important thing. And there's this quote from Linus, the Linux guy, right? Given enough eyeballs, all bugs are shallow, right? Enough people look at code, the bugs are going to be found. And there's a nice paper by Petrie and Wilson from a while ago. It's basically about how to run a code review, for science. We try to do this in my lab regularly, where we sit down in one of our lab meetings and somebody has a piece of code they want to walk through. And it's not so much about like, oh, let's go look for bugs. It's really about how the way we usually do it is they give me the code, we put it up on the screen, and I start reading through it, or whoever's running the review starts reading through it and basically just ask questions. Why did you do this? What are you doing here? What does this mean? Just to try to make clear, like, what are the questions that one might have that you can't answer just by looking at the code? Because ultimately you want the code to be able to answer all the possible questions, either through its structure or through judicious comments. Now, fortunately, the AI tools have become really good at reading code and commenting on it. I'll talk some more about that tomorrow. So software testing is an important thing. It's also kind of a pain in the butt often. So I certainly don't write tests for all of my code. I try to write tests at least for the really important things. For anything that we're reusing in the lab, we write tests for, because we don't want something to be reused by a lot of people and then realize that it's wrong. There's various different types of tests. Some of them are just like, does it run without crashing? We call that a smoke test. Does it start smoking? Sanity checks, I'll show an example of this. Are the values within reasonable ranges? Are there any negative reaction times, for example? Those shouldn't exist. And then what most people in computer science focus on, or in software engineering focus on, are what we call unit tests, which are basically looking at each module, like each function, and basically saying, is it doing the right thing? Is it giving the right answers? Is it doing what it's supposed to do? This is an example of where doing a little bit of testing could have really saved somebody. A paper was published in PLOS One that had to be corrected after somebody realized that the data included two notable age outliers. It was supposed to be adults, right? And they had somebody age 5 and somebody age 32,757 years. Clearly not a reasonable age for a human, at least not in years. And so they had to change when they fixed it, it actually changed the results. So they had to report these differences in the results. What they could have done is, let's say that subjects are supposed to be between ages 12 and 120. They could just put this one line in their code, similarly, that basically says, if this statement is not true, then raise an error. That's what an assert does. This is in R, but similar thing you could do in Python. No, actually, sorry, this is Python. So, anytime there's something that should be true, like there should be no negative reaction times or 0 reaction times, there should be nobody whose age is outside of these particular ranges, things like that. You should put those in. Another place where I always try to put asserts in is when I'm changing the shapes of data. And I always want to make sure that the new thing that I've generated, so let's say I have 2 different things that are supposed to be, have the same number of rows. I always want to make sure that they have the same number of rows, just in case some, other piece of software down the workflow tries to reuse them and things aren't matched up. Anytime you have like, things with IDs, like subject IDs or something like that, always making sure that the IDs are lined up because sometimes, let's say that the IDs get missorted in one data set versus another, but you're assuming that they're the same. So you should always make sure anytime there are like keys to a data set and you have two different versions, make sure those keys match. So things like that, where you should always be putting in these little asserts. They're really easy and they'll save you a ton of potential pain down the road. There's an idea that I wish I could incorporate into my workflow. I've tried on occasions. Other people in the lab have been more effective at it, called test-driven development. The idea is that rather than writing code and then writing tests to make sure it's doing the right thing, you basically write the test first that will check and see if the thing is doing what it's supposed to do before you ever wrote the code to do it. So the tests are written and they're going to fail the first, they have to fail the first time that you try to run them. If they don't, then you've done something wrong. And once the test passes, you're done. You can go back and refactor it to make the code better or more robust, more modular or something. But the idea is that having a test for the functionality you want can, one, tell you once you've achieved that functionality, and also prevent you from writing a bunch of code that you don't need and that's not going to necessarily be tested. So I like, at least in thinking about how you go about writing code, I think thinking about things like test-driven development are really useful, especially if there's anything that, like any functions you write that are being reused a lot. You might start by writing tests for those and doing this test-driven development thing, even if the whole workflow doesn't end up being kind of fully tested. I don't know if you will, in the next 2 weeks, encounter automated testing, but it turns out that it's very easy to, once you have tests, it's very easy to have those tests be run in an automated way via GitHub. GitHub has something called GitHub Actions, and very easy is relative, right? It takes a little bit of work, but they make it as easy as they can. And so the idea is that if you have a set of tests in your GitHub repo, you can basically, every time you push from your machine to the repo, it will automatically run the tests to make sure that, really to make sure you haven't broken anything, or to make sure that things are working. It also really helps because it makes sure that things will run on a different system than your own. Now, this will break down if you're working with huge data sets. You might have to build like a tiny test data set. That's what we, like for fMRI prep, which is our pre-processing workflow, we had to build like small test data sets because we can't upload huge data sets onto the test platform. But this, you can't see this, but the idea is like, the GitHub will help you write a script that basically then tells the test how to work. I have an example. I have a PyTest tutorial on my GitHub account that you can have a look at, and it shows how to do this. And then this is what you see when you go, let's say I did a merge cleanup and I broke something. Now it's going to give me the red X and say, oh no, it broke, the tests failed now. So this can be really useful as you're developing code to kind of help you just get into the habit of always testing things. And it's nice because it can keep you from breaking things that used to work, or at least tell you, won't necessarily keep you from doing it, but it'll tell you if you're breaking something that used to work. So I'm going to end just with a couple comments on a really important distinction, right? I've focused here on reproducibility, which is just like getting the same answer, really. And in psychology, we talk about the distinction between reliability and validity. So you can really think about reproducibility as like the reliability concept. Can you get the same answer each time? Can you run the same code each time and get the same answer? But we want our code to be both reliable and valid, right? We want it to get the same answer each time, but I can get you the same answer each time by just having it spit out a bunch of zeros, right? It's not the right answer, but it's a consistent answer. We want the answer to be right as well as being reliable. So that's captured by this distinction between, we want it to be both reliable in the sense of having a, low variance, but also be valid in the sense of like actually hitting the target that we want to hit. And so one thing that I think you should think a lot about when you're writing code is also, how do you validate the code? How do you make sure it's getting the right answer? Not just make sure that it's like, doing the right computation, but getting the right answer at a larger scale. So how do you do that? Well, one thing you can do is I think learning how to simulate things is a really important skill. You're, wherever you are in your career. I find it super useful all the time because as somebody who, there's a great quote. It's to the effect of like, kind of decent skill at simulation is generally going to be much more effective than knowledge of a ton of math, right? Like whenever I can't understand something, usually a piece of math, the first thing I try to do is go simulate it, because that's a really good way to understand. Or if something's not working, model's not fitting, before I do it with real data, I write a small simulated data example just to help me figure out what's going on. So one thing you can try to do is what we call parameter recovery, is like create some data with a known answer and then make sure that your code can get that answer back. People do in the kind of computational modeling area in psychology do this all the time, but I think it's a useful tool in general. Another is, what we might call null or randomization testing. So if you, this is really important if you're doing any kind of cross-validation, is to basically put at the very highest level, take your data set and randomize the thing you're trying to predict across subjects or across trials, whatever it is you're trying to randomize. And make sure that if you do that a bunch of times, the answer it gives you is that there's no effect there. If those people with a suicide paper had done that, they would have seen that, hey, even when the data are random, we're getting really good results. And that would have been a signal. And there are increasingly powerful frameworks for generating data. This synthetic data vault thing is one. They're not easy to use. There's something called fMRI sim for fMRI data, really not easy to use. Hopefully they will get better. But I think learning how to even do simple simulations is a really powerful tool. Okay, so I hope I haven't demoralized you too much. The goal is to really to give you motivation to spend the next 2 weeks figuring out how to not get stuck in these same problems, right? And I think that, there are things that are in your hands and there are things that are out of your hands, right? Getting large enough data sets might not be in your hands, but learning the skills to do the best possible job of data analysis that is something that's in your grasp. And so hopefully you'll all learn a lot in the next 2 weeks about how to do this stuff. So thanks. Yeah.
It's kind of related, but we're wondering nesting functions. I tend to nest functions that I don't use. And I'm just wondering whether
I think it can be good or bad. I don't think it's intrinsically good or bad. I think that, so the principle that, to the degree that it drives you to adopt the principle I'm about to state, I think it could be a good thing, which is that a function should do one thing, right? A function should do a thing where you can say, this function is doing this thing, right? If a function is trying to do too many things, then it's better to break that into separate functions. And sometimes those are going to need to be nested depending on the structure of what you're doing. So I think that making, anytime a function is more than, 50 lines for sure. I like the rule, a function should be something that you can kind of see on the screen, right? If you can't, sometimes you're going to have bigger functions, but often what that suggests is you need to modularize things to break stuff out into if you have a loop within a bigger function, often you want to break that out into a separate thing. So I don't know, does that address your question?
Yeah.
Cool.
Question on Zoom from Angelina.
Cool.
Wait, let me just get this right. Yes, I'm here now. Sorry.
Cool.
Yeah, thank you. Thank you so much for this part of the lecture as well. Very fascinating. So again, I'm trying to approach this from the perspective of a PhD student. I feel like PhD students oftentimes do not have a lot of supervision when it comes to data analysis. So I feel like many of us who are not programmers end up having to learn some coding and finding their own way to orientate themselves in the world of programming. And I'm wondering, for people who are not programmers but have to code for their academic work early on, would you typically suggest using tools like ChatGPT? What's your opinion on this? Because, for instance, I use it all the time. I feel like I'd be quite helpless without it, really.
Yeah, no, I think that the AI tools, and I'll say more about this tomorrow, but I think that they are really helpful for people who are just getting started. Because often, the code that they generate is usually actually pretty good, certainly compared to what a really novice coder might write. And so I think that they can often help one get past, so there's like there's a few reasons that coding is difficult to learn, right? One is coding is really it's ultimately about solving problems, right? And so there are strategies for solving problems that you have to learn to write code. But then there's also remembering all the details about how you actually write the thing down, right? So there's there's the ideas and then there's the language. And the AI tools are like, if you have an idea of what you want to do, but you just don't know the Python syntax for it, the tools can be really good for that. And then hopefully you will learn over time, how to leverage them in such a way that, that you learn, by working with them, how to actually write the code and how to read the code and see what it's doing. And that ultimately you'll be able to, kind of write in a more effective way yourself. I mean, I don't think, to foreshadow what I'll say tomorrow, I mean, I think everybody should be using AI tools to help write code. Like if you're not, you're basically wasting time and you're probably making coding less fun than it would be otherwise, right? Because there's a lot of things that these tools can do that just address like stupid drudgery that there's 20 lines that need to be written. You know what they're going to say. And it's just like, can I write them all without having any typos, right? Whereas I can just put a comment that says, I want to do this here, and it'll write those for me because they're so kind of obvious that, that the algorithm just knows what they're doing. And that just makes programming a lot more fun because it frees you up to think harder about the actual, conceptually important and difficult parts of coding. Was that a helpful answer, or is there anything else I can say?
No, that's definitely reassuring because sometimes I end up feeling the exact same way. What if everything I've done is absolutely wrong? And what if I end up publishing that paper? Because I do check my code a lot. And typically I would rerun it on different data sets, but say sometimes I wouldn't find an effect. And then I would think, well, did I check the distribution of the data? Maybe I had to log transform it or do something else with it. And then I end up questioning my decisions and thinking this is probably all wrong. And this is just fake science. And I'm generating it without knowing because I'm doing this to the best of my ability. But I only have ChatGPT and Google to fall back on. It's not like an experienced researcher or programmer is checking my code each time I have to write something. So I feel like sometimes as a PhD student, especially, you can feel quite lonely and intimidated because essentially you just have your own devices and whichever way you found to work around big issues with code. So yeah, I think it's a very real problem for many of us, really.
Yeah. I mean, the other thing that you can think about doing is, finding people from other labs or even other departments, on your campus who are struggling with the same problems. I think in most, most places there's right now across many of the sciences, there's a lot of people, starting to learn Python, doing it using, AI assistants, and probably struggling with a lot of the same problems that you are. So even if in your lab or in your department, those people don't exist, they almost certainly do across campus. And then the question is just, how do you find them? And so,
or what do they want in return? Because sometimes also authorship becomes a problem where if you get someone to check your code and partake in a study, then, should they also get authorship? Because you want to give credit where credit is due. So I personally try not to not give credit if someone has actually helped. But I think then that becomes a bigger problem really with adding more people.
Yeah, I agree. I think it's worth having those conversations up front about what's what's the bar for authorship on a particular project.
I just want to add that
Thank you.
You can probably look for those people at the Neural Academy here.
Yeah, no, this is, you're building a community by coming to this. Yeah.
There's a couple more questions online. We probably just have time for one more question before lunch. Anyone else who has other questions, please post them in the Slack channel for the topic. We keep talking about this. But I think Alex had his hand up on Zoom first. So maybe Alex, go ahead and ask your question.
Sure. Yeah, I just had a quick question. What do you think about the trade-off with larger sensor arrays or the ability to collect more data as far as brain-wide, smaller voxel sizes or something like that with reproducibility and correcting for that?
Sorry, I missed, you said the trade. I didn't catch exactly what you said about the trade-off. You're talking about moving to 7T and higher field strength and higher resolution. Is that what you're getting at?
Yeah, is that going to make us need, does that increase the scale then more because we have more comparisons, if everything is, if we're not using a model?
Yeah. I think, there's probably pluses and minuses, right? I think that it has to, the data needs to fit with the hypothesis, right? If you want to ask hypotheses that are about things that are at a neural resolution that, let's say you want to go find, particular types of columns, or you want to do laminar analyses to ask about, deep versus shallow layers of cortex. Those you just can't really do effectively at, with lower resolution or lower field strength. So there are going to be times when you really need that higher resolution. And I think that then the question becomes, what's the right way to analyze those data that addresses the potential challenges around, for example, the fact that you're going to have a lot more voxels, so you have a lot more comparisons to be done. So I don't think it's intrinsically good or bad. I think it's going to, that it just requires that one bring the right tools to bear. Though, I mean, the one thing I would say is right now, kind of high field imaging is still pretty much the Wild West in terms of how data are being processed. I'm hopeful that we can, for example, we want to bring, like fMRI prep doesn't really work with 7T data right now. We'd like to make it work with 7T data, something we want to do in the future. And I'm hopeful that in time that field will kind of, develop a more kind of constrained set of practices.
Sure. I just think as a thought experiment from a reproducibility perspective, if you have, 4 brain voxels, then you only have 4 tests, and, maybe you have more positive findings because of that. Whereas if you have, you do a Bonferroni correction and you have, 4 million voxels, you'll correct out most things. So in some way, there's a perverse incentive to have lower resolution with
Right.
class.
Yeah, and there are, but there are also tools that one can use to try to, I think, help address that challenge in a way that isn't as draconian as something like a Bonferroni. So I think ultimately that, one should think about like what's the signal in the brain that you're trying to find, and what's the most appropriate measurements to find that signal. And if it needs to be high resolution, then it should. If it doesn't need to be, then it shouldn't.
Sure. Thanks. Yeah. Interesting.
Yep.
Hi, I have a question about synthetic data. As you have mentioned that synthetic data may be used to make more validated code and may be used for validation. But I have a query about that. These simulated data or synthetic data are actually generated on the basis of actual data, and these models try to learn about the distributions of the actual data from these samples coming from. But my question is, how do you validate that these generated data points come from the variation they come from the original data is not much, or they actually correspond to some sample from the original distribution?
Yeah, no, I think
Because
there are serious challenges about exactly how to use real data to generate simulated data, because you certainly don't want the joint distributions in the original data to affect the joint distributions in the simulated data, because then you're basically peaking at the original data. So we usually think about, just generating kind of pretty simple simulated data, just using like simple assumptions of, Gaussian noise and things like that, that look like the original data in terms of their shape. But we generally have avoided using the real data to generate simulated data in anything more than that kind of like really dumb way, because of concerns about exactly this, about like leakage of information from the original data into the simulated data. But thanks for the question.